Part 1. Please find a quiet place to sit or lie and to listen to this audio. Once seated or lying, close your eyes. Focus your attention on your breath. When you're ready, inhale slowly. Let the air fill your lungs. And then exhale slowly. Let the air escape from your lungs. Inhale again. And then exhale again. Notice how your stomach rises as you inhale and falls as you exhale. Rises as you inhale and falls as you exhale. If your mind wanders, don't be concerned. Just gradually shift your attention back to your breath. Now, for a moment, focus your attention on one foot. What sensations do you feel? Do you feel a tingling, a lightness, a warmth, or anything else? You do not need to create these feelings. Just be aware of any sensations that you might feel. Now shift your attention to the calf just above this foot. What sensations do you feel now? If you do not feel any sensations, don't be concerned. Just feel aware of the absence of sensations. Now shift your attention to your thigh, just above this calf. What sensations do you feel now? Does your thigh feel warm and relaxed? If your thigh feels tense, you can just observe this tension. Or as you inhale, you can imagine drawing this tension into your lungs and then exhaling this tension out into the air. If you like, you can stop this audio for a while and continue these exercises with your other leg and then your hip, your stomach, your back, your chest, your arms, your hands, and your neck. Or you can continue this audio now. Now shift your attention to the cells in your body. The human body contains about 37 trillion cells. Some cells are circular, like footballs, and some cells are long, like spaghetti. But imagine just one of these cells, perhaps a cell on your skin or in your brain. Inside this cell, like inside every other cell in your body, is a large, dark sphere. This large sphere, called a nucleus, is like the core of an apple. Now imagine your attention closer to this sphere. Imagine you can see inside this sphere or nucleus. In this nucleus, as in every nucleus in the human body, imagine 23 pairs of spools, similar to the spools, reels, or the bobbins around which you would wind cotton. Of course, the nucleus does not actually comprise 23 pairs of spools, but just for a moment imagine the cells comprise these 46 spools, arranged as 23 pairs. Part 4. Now zoom your attention onto one of these spools. As you zoom your attention, you realise that a thread seems to be wrapped around these spools. Now imagine mentally pulling some of this thread away from the spool, perhaps with a sewing needle. Imagine a loop of thread is now pulled away from the spool, rather than wrapped around tightly. As you look closely at this thread, you're surprised. This thread looks different to what you imagined. This thread actually resembles a long, thin, winding ladder. That is, the thread actually contains two parallel strands connected by many small rungs. The two parallel strands, that is, the sides of this ladder, are composed of a sugar called deoxyribose, interspersed with specific molecules called phosphates. Don't be concerned if you can't remember the name of this sugar or the name of this phosphate. The details aren't important. But the image of a soft, winding ladder wrapped around a spool is important. Fortunately, the rungs of this ladder are more interesting. Each of these winding ladders comprises about 50 to 100 million rungs. Imagine that each rung comprises two squares, like two tiny squares of cloth or paper, are joined alongside one another. and each square comprises one of 4 colours: red, green, blue and yellow. So for example, one rung might comprise a red square alongside a green square. The next rung comprises a blue square alongside a yellow square. The next rung comprises a green square alongside a red square. And the next rung comprises a yellow square along a blue square, and so forth. But something about these rungs seems unusual. You scan this sequence of colours for a while, uncertain why these rungs seem unusual, but then suddenly the answer bursts into your awareness. In particular, you notice that one yellow square always accompanies one blue square, and similarly one red square always accompanies one green square. So some rungs comprise one yellow square and one blue square, like the yellow sun affixed to the blue sky above. And the other rungs comprise one red square and one green square, like the top and bottom of a traffic light. Now actually, these rungs don't really comprise blue, yellow, red and green squares. These rungs actually comprise 4 chemicals: adenine, cytosine, guanine and thymine. And each rung comprises either one adenine and one thymine, or one cytosine and one guanine. But for our purposes, you don't need to remember these chemicals. Just focus on the coloured squares, blue, yellow, green and red, that represent these 4 chemicals. Indeed, you don't need to devote any effort into this exercise. You merely need to follow the instructions. Now this long winding ladder, wrapped around a spool in which each rung comprises 2 coloured squares, is called DNA, or deoxyribose nucleic acid. Each of the 46 ladders, one corresponding to each spool, is called a chromosome. And the spool itself is called a histone protein. As you visualise this spool, the spool around which the DNA or ladder is wrapped, just repeat to yourself, histone, histone, histone. However, if you don't remember this term later, don't be concerned. This term is not vital for our purposes. You've probably heard about DNA and chromosomes. You have perhaps known that DNA comprises a code that differentiates every human, and indeed every animal, plant, and even bacteria. Part 5. But how does this DNA differentiate people? After all, every human, the DNA is primarily wrapped around a spool or histone, and in every human, the DNA resembles a coiled ladder in which each rung comprises one adenine and one thymine, or one cytosine and one guanine. And in most humans, but not animals, the number of chromosomes is 46. So how do humans then differ from each other? The answer is primarily that the order of colours varies across humans. So to illustrate, imagine you could detach the winding ladder from one spool, perhaps the smaller spool. And imagine you could divide this ladder into 2 halves, the left side and the right side, so that each half comprises one rung. You could now describe the sequence of rungs on the left side, beginning with the first rung. The sequence might be red, then yellow, red, green, red, blue, yellow, and so forth. Now suppose you repeated this task, but using the DNA from another person. The order of colours would differ. Beginning with the first rung, the rungs on the left side might be blue, green, green, yellow, red, red and yellow instead. Now, of course, you don't have to describe all 50 to 100 million rungs. Even after you describe just a few rungs, you might understand how the DNA of one person differs from the DNA of another person. You'll appreciate how the order of these 4 colours, or more precisely the order of these 4 chemicals, differs across individuals. Part 6. Scientists tend to divide these 50 to 100 million rungs or so on each chromosome to about 20,000 subsets called genes, and each gene comprises thousands of rungs. For each gene or segment of DNA, the first 100 or so of these rungs look similar in each gene. For example, these rungs might comprise a particular sequence of colours on the left side, such as blue, red, red, yellow and blue many times. And this section of the gene is called the promoter region. And as you visualize this first section of the gene, repeat the term promoter region, promoter region, promoter region. Later you will discover this region is vaguely like the on switch of each gene. The next few hundred or even 1,000 rungs differ markedly across the genes, and this second or middle section is called the coding region. As you visualize this second or middle section of each gene, repeat the term coding region, coding region, coding region. Later, you will discover this region generates the codes that produce the proteins that ultimately become who we are. The final or so 100 rungs also look similar in each gene. Perhaps for each gene, the final 100 or so rungs on the left side comprise a sequence of yellow, 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 blue and red many times. And the final section of the gene is called the terminal region. As you visualize this final section of each gene, repeat the phrase terminal region, terminal region, terminal region. Later you will discover this region is vaguely like the off switch of each gene. Admittedly, this description is a simplification. For example, genes do comprise other regions, and some of the winding ladder, the DNA, does not comprise genes. But you don't need to concern yourself with these complications now. Part 7. But how do these genes produce the proteins that ultimately become who we are? Well, to answer this question, suppose that you pulled some of the winding ladder of thread, in particular one gene, away from the spool. Now imagine that you watch a molecule swim towards this gene, and perhaps assume this molecule resembles the top section of a zipper, that is the tab of a zipper. This molecule is called RNA polymerase. As you observe this molecule approach the gene, repeat the term RNA polymerase, RNA polymerase, RNA polymerase. Now imagine this RNA polymerase somehow binds to the promoter region. Once attached, the RNA polymerase, the tab of a zipper, in essence activates the gene. The gene is switched on. In particular, once attached to the promoter region, imagine the RNA polymerase descend down the ladder past the coding region. As the RNA polymerase passes a specific rung, the two squares in this rung are yanked apart. For example, imagine the red square and green square of the first rung are pulled apart. And then imagine the yellow square and blue square of the second rung are pulled apart. And next imagine the third, fourth, fifth, and indeed hundreds of other rungs are yanked apart in sequence. Part 8. In the end, the coding region of this gene is separated into two sections, the left side of the ladder and the right side of the ladder. But why does RNA polymerase separate the two sides of this coding region? What is the purpose of this change? You soon notice that other squares will then bind to the left side of this ladder. That is, the complementary colour will bind to the left side of these rungs. For example, imagine the first rung in the left side of this coding region is red. So a green square will then attach to this red square. Imagine the second rung in the left side of this coding region is blue. A yellow square will then bind to this blue square and so forth. And so perhaps if you can, continue to imagine other squares gradually attached to the left side of this coding region. Therefore, as the RNA polymerase plunges down the ladder and separates the left and right sides, another series of squares will develop. And this series of squares will be the same as the right side of the winding ladder. Eventually, the RNA polymerase will arrive at the terminal region of this gene. The RNA polymerase will then detach from the ladder. As this RNA polymerase drifts from the ladder, the molecule drags the new sequence of squares in the same direction. Perhaps you can visualize this sequence of squares, red, blue, green and yellow, drifting away. And this sequence of squares is called messenger RNA, the reason for which might become clearer soon. As you visualize these squares drifting away, repeat to yourself, messenger RNA, messenger RNA, messenger RNA. Part 9. But what is the purpose of this messenger RNA? At first glance, this messenger RNA does not seem especially useful. After all, this strand of squares appears to mirror DNA, or in particular, the right side of this winding ladder, or at least a segment of the right side of this winding ladder. Besides resembling only one side of the winding ladder, this messenger RNA differs from the DNA on 3 attributes. First, when you imagine this messenger RNA closely, you might notice that one of the colours differs from your expectations. For example, you might notice that the red on the DNA is darker than is the red on the messenger RNA. Now, of course, these colours are actually just metaphors. To be precise, 3 of the chemicals, adenine, cytosine and guanine, are the same in DNA and RNA. However, DNA comprises thymine, whereas RNA comprises uracil. Although almost the same, the chemical on RNA is not as stable or enduring as the chemical on DNA. You can imagine the light red squares that are representing the chemical on RNA tearing or dissolving after a few minutes. Second, for both DNA and messenger RNA, the sides of these letters comprise a sugar. And this sugar is called deoxyribose for DNA, but ribose for messenger RNA. Now, these 2 sugars are almost identical, except ribose contains an extra OH, or oxygen and hydrogen, at one end. Because of this extra oxygen and hydrogen, the sugar on RNA is not as stable or enduring. Now, don't worry if you don't remember the details, like the name of uracil or the name of these sugars. Just remember that messenger RNA comprises chemicals that are not as stable or enduring. And you might wonder why messenger RNA is not as stable or enduring as DNA. Has nature committed some sort of error? Or is messenger RNA deliberately unstable? Well, the answer is that messenger RNA is deliberately unstable. In particular, whereas DNA is designed to last a long time, usually your entire life, messenger RNA is designed to last only fleetingly. In particular, as you will soon understand, messenger RNA is designed to last only long enough to create the necessary proteins. Part 10. To clarify, messenger RNA drifts from the nucleus, the dark core inside every cell, to a region of the cell outside the nucleus. Perhaps you can imagine this long structure resembling one half of a winding ladder departing from the nucleus. Once outside the nucleus, visualize a series of molecules like tiny ribbons surge towards the messenger RNA. You then notice these ribbons bind to the messenger RNA. In particular, each ribbon seems to bind to a sequence of 3 rungs. One ribbon might bind to the first 3 rungs of this RNA. A second ribbon binds to the next 3 rungs of this RNA. A third ribbon binds to the next 3 rungs of this RNA, and so forth. The ribbons don't look identical either. Some ribbons are slightly longer than other ribbons, some ribbons are curlier than other ribbons, and so forth. And then you notice the ribbons can be divided into 22 distinct shapes. If possible, try to visualize 22 or so distinct shapes or categories of ribbons. Perhaps one category of ribbons resembles little coiled worms. Another category of ribbons might resemble long streamers, and so forth. Then you realise that each category of ribbons binds to a particular sequence of 3 colours. For example, the coiled worms might always bind to a sequence of red, green and blue squares. And the long streamers might always bind to a sequence of yellow, yellow and red squares, and so forth. Now, you're now closer to understanding the purpose of messenger RNA. But you need one more chunk of information, and that is that these 22 categories of ribbons actually represent 22 distinct amino acids, the building blocks of proteins. So now you might be able to gradually integrate this information to understand how DNA generally generates proteins. In particular, DNA, that is the winding ladder, wound around a spool, produces these segments of messenger RNA, a sequence of colours. And then amino acids bind to each sequence of 3 colours. And each combination of 3 colours, that is actually 3 chemicals, represents a specific amino acid. For example, the sequence red, red, blue might attract the amino acid tryptophan, and the sequence green, blue, yellow might attract the amino acid lysine, and the sequence yellow, blue, and red might attract the amino acid glycine, and so forth. So hence, each messenger RNA, and thus each gene, produces a particular sequence of these amino acids. And a sequence of amino acids is called a protein. So therefore, each messenger RNA, and thus each gene or segment of DNA, actually produces one protein. Part 11. So this description offers some insight into how genes produce proteins. But how are these proteins converted to an entire human? Surely our body and brain comprises more than merely proteins. Surely we comprise many other chemicals as well. Actually, these proteins serve 2 main functions. Firstly, our cells are largely composed of proteins. Therefore, these genes produce some of the main features of our cells. But secondly, proteins are also enzymes that facilitate the formation of other chemicals. Part 12. But one question, or at least one question, remains unresolved. You might have heard that every cell comprises the same DNA, the same genes. So every cell should produce the same proteins. Therefore, every cell in our body should look identical. We should look like one mammoth blob of identical cells. Yet actually, at any time, not all of our genes produce proteins. That is, genes that are wound tightly around the spool or histone seldom produce proteins, they're inactive. Whereas genes that are not wound tightly around the spool or histone tend to produce proteins. Many events and chemicals affect whether or not a gene is active. To illustrate, many activities from quarrels to hugs affect the concentration of chemicals in our body and in our blood. And these changes even affect the concentration of chemicals in the nucleus, the dark sphere in every cell that houses the spools of DNA. And chemical changes in the nucleus can affect which genes produce proteins. So to illustrate, in response to these chemical changes, imagine small molecules, perhaps resembling tiny M&Ms, attach themselves to the histones, the spools around which the DNA is wound. Now actually, these small molecules are called acetyl groups and comprise a sequence of carbon, oxygen, carbon, and 3 hydrogen atoms. And actually, these small molecules, resembling tiny M&Ms, bind to the thin tails that are actually attached to the histone. But these details aren't important. Instead, you merely need to know that in response to events, various chemicals, like acetyl groups, might become more likely to bind to the histone at particular locations. And at these locations, the histone does not pull the DNA as tightly. The DNA is separated from the spool or histone. Hence, the genes at this location can more readily produce proteins. Now, so you don't have to remember the details of this example. Simply note that events in our lives affect the concentration of chemicals in our body, and these changes can affect which genes produce proteins. Likewise, after other events, another molecule called a methyl group, or CH3, often binds to the promoter regions of genes. And these methyl groups tend to inactivate the gene. We do not yet know precisely which events affect the likelihood that acetyl groups, methyl groups, or other chemicals activate or inactivate genes. But we do know that many events do determine which genes produce proteins. Part 13. But more remarkably, events in our lives do not only affect which of our genes produce proteins, but can affect future generations as well. So to illustrate, traumas in our life might increase the likelihood that methyl groups bind to genes in many cells of our body. To illustrate, trauma might increase the likelihood that methyl groups bind to genes in our sperm or ova. Remarkably, when these sperm or ova produce zygotes, and eventually a fetus and baby, these methyl groups are still often bound to the same genes. In other words, events in our life can affect which genes are active in our offspring. That is, our experiences can affect which proteins our babies produce. Our experiences can change our babies.